Imagine a world where our crops can withstand the harsh realities of climate change. This is not just a dream. It's becoming a reality thanks to groundbreaking research led by an international team from Heinrich Heine University Düsseldorf, HHU, and the Max Planck Institute for Plant Breeding Research in Cologne, MPIPZ. They have unveiled a cutting-edge method for pinpointing what are known as genomic regulatory switches, tiny yet powerful elements that dictate how plant traits are expressed. In their recent publication in the esteemed journal Nature Genetics, the researchers reveal that although these regulatory switches constitute a mere fraction of the entire genome, their impact on plant characteristics can be profound. Their innovative approach has already shown promise in identifying key switches related to drought stress, paving the way for the development of new maize varieties that are better suited to the challenges posed by climate change. Biodiversity, driven by natural genetic variation, is essential for evolution. However, the slow pace of natural evolution, often taking thousands of years, leaves us at a disadvantage in adapting our crops to the rapidly shifting climate, which is leading to more frequent droughts. To ensure global food security, it is crucial for scientists to expedite the discovery of beneficial natural DNA variants that can enhance crop performance under stress. In a significant advancement, Dr. Thomas Hartwig and Dr. Julia Engelhorn from the Institute for Molecular Physiology at HHU and MPIPZ have introduced a new, efficient technique for mapping the genetic switches in plants, as detailed in their latest article in Nature Genetics. These switches, while not genes themselves, play a critical role in determining when, where, and how much a gene is activated. Think of them as dimmer switches that control the brightness of a lamp, adjusting the light according to need. Historically, research has concentrated primarily on the genes themselves, but this new study highlights that many of the key differences observed between plants, such as variations in size or resistance to diseases and stress, are often dictated by these regulatory switches rather than the genes. Traditionally, pinpointing these regions has been a challenge, as has identifying which specific changes are most impactful. However, this new scalable mapping method developed by the research team is set to change that. The researchers examined 25 different maize hybrids, which are crossbreeds of various maize varieties, and discovered over 200,000 regions in the genome where natural variations affect these regulatory switches. Dr. Julia Engelhorn, the lead author of the study, states, "Even though these regulatory switches account for less than 1% of the genome, the variations can often explain a significant portion of heritable trait differences, sometimes more than half." Dr. Thomas Hartwig, the corresponding author, adds, "By understanding how these regulatory switches function, we gain a powerful new tool to improve both crop resilience and yield, setting the stage for more intelligent breeding practices in the future." The team specifically focused on traits related to drought stress, identifying over 3,500 individual regulatory switches and the corresponding genes that enable plants to adapt to water-scarce conditions. Engelhorn explains, "Our method allows for a direct comparison of the differences in switch variants inherited from both maternal and paternal lines within a single experiment. This provides the maize research community with a valuable resource of over 3,500 drought-related regulatory sites, opening new avenues for fine-tuning gene expression to enhance plant robustness." Hartwig emphasizes the significance of this precise mapping. By learning from the natural variations in these switches, we can understand their mechanisms, which in turn allows us to manipulate them intentionally to create plants with improved traits. This research was conducted in collaboration with a team from the University of California, Davis, including Dr. Samantha Snodgrass. As a co-author of the study, she highlights a shift in perspective that this approach brings. Despite decades of successful research, much of the genome, specifically the regions outside of the genes, has remained a mystery. This new method sheds light on these non-coding areas, providing biologists and breeders with precise targets for innovative research and development strategies. The study was part of the C+ Cluster of Excellence on Plant Sciences at HHU and MPIPZ, with additional funding from the European Horizon Europe project, Booster, which aims to promote the development of climate-resilient cereal crops. What do you think about this new approach to crop breeding? Could it be the key to ensuring food security in a changing climate? Or are there potential pitfalls we should be wary of? Share your thoughts in the comments.